to introduce Florence Dubois-Brissonet from AgroParisTech. She is professor in microbiology and food safety. She was working in AgroParisTech since 1992. Since 2012, she got the status of assistant professor. No, sorry, associate professor, full professor. Her research is on biofilms and hygiene, and also she is leading a research team on this topic where she is working on different issues. She is also involved in teaching, so she is co-director of a master degree program in food safety risk analysis and also coordinator of a number of graduate programs on this topic. So with this, I would like to finish, and I will give the word to Florence to start with the presentation. I will make her now presenter, and she has now the control to show us her presentation. Florence?
Yes.
Hello, yeah.
Yes, it's okay?
Yes, perfect.
Okay, so thank you very much, Gerhard, for this introduction, and thank you to all the participants for attending this webinar. So the topic today is about emerging microbiological hazards and how they can bring new challenge to food safety. So despite food law regulation requires from food operator that they shall not place unsafe food on the market, there are still many foodborne outbreaks in Europe and all over the world. For example, in 2013, there were more than 5,000 outbreaks and 40,000 cases in Europe. So why? Mainly because of the mistakes. So mistakes because of contaminated raw materials, or mistake during food preparation, or compromised condition of preservation. So this is very well known. But sometimes some emerging risks can appear, and then we don't have relevant control measure. So an emerging infectious disease can be a new disease, an unknown disease that emerges among the population, but it also can be a rapid increase of incidence or dissemination of an already known disease. All the factors that leads to emergence of microbiological disease can generally be classified in three parts, in three categories. The first category is about microbial adaptation. The second is about the change that can be done in microbial environments that can lead to difference in transmission of microorganisms to the human being. And the third part is about human susceptibility. The time scale of emergence can be very different depending on the case. Sometimes the change is very low, low speed, very slow, sometimes notably when it's due to climatic changes. But sometimes it can be very quick, very rapid, for example, when a plasmid is transferred. Similarly, there is a very different spatial scales for emergence of microorganisms. Sometimes the area is very restricted, just like for plasmid transfer, or it can be very large when globalization of trade is concerned. So we will take some example in these three categories for microbial adaptation, for changes in microbial environment, and for human susceptibility. But before that, just a bit of history. the major epidemics before the 19th century were eradicated before the discovering of antibiotic, with disinfection of drinking water, with sewage treatment and milk pasteurization mainly. And with in the 50s, also the refrigeration was really important, because it was a very important technology for food preservation. But after that, some new microorganisms emerged. For example, psychotrophic bacteria were very important because of the generalization of refrigeration. The main example are Yersinia enterocolitica, Listeria monocytogenes, or Clostridium botulinum type E. Moreover, some zoonotic agents also emerge. Zoonotic agents are transmitted from animal to human. For example, Campylobacter, E. coli, enterohemorrhagic E. coli, or Vibrio parahemolyticus are zoonotic agents that emerge in the second half of the 20th century. So now some examples in the three categories. The first one is about microbial adaptation. So a new pathogen will appear. It can be a newly identified pathogen. So for example, when we discovered the parasites Cyclospora, it was not identified before, and it appears in an outbreak. We didn't know what it was, this microorganism. So it is newly identified pathogen. But sometimes it can be a pathogen that is already known, but it becomes more virulent or more resistant because it adapts or it takes some new characteristic. So this adaptation can be whether genetic changes or phenotypic changes. The first example is the new virulent factor that can be acquired by bacteria by horizontal genetic transfer of gene. A good example for that is E. coli enterohemorrhagic O157H7. This E. coli has a SIGA toxin, and it causes hemolytic and uremic syndrome in children. And generally, the food that is associated with this outbreak are uncooked or poorly cooked minced beef. This SIGA toxin producing E. coli comes from a non-toxinogenic ancestor. It evolved by horizontal transfer of gene. Two genes of coding for SIGA toxin, STX1 and STX2, were transferred to these bacteria by a bacteriophage. So this bacteria was more virulent than the its ancestral strain. In the 2000s, other enterohemorrhagic E. coli appeared. For example, here you see in orange all the new strain that appeared from 2000. One example is the E. coli O104H4 that caused an important outbreak in Germany in 2011. This outbreak concerned mostly adults, which it was really unusual for enterohemorrhagic E. coli, because generally it's for children. But this can be explained because this strain is an enteroaggregative E. coli, and this strain acquired the gene coding for the SIGA toxin by horizontal transfer. So this was a new pathogen. This explained because that the epidemiologic investigation was very long and difficult, and at the first time the cucumber was suspected, but at the end it was found that the origin was fenugreek fruit seeds. Besides genes of virulence, there are also some genes of resistance that can be transferred from a bacteria to another. Notably for the zoonotic agents that are constantly in contact with some antibiotics. A good example for that is Salmonella, because the global prevalence of Salmonella in food decreased in France and also in Europe, but there are some new multidrug-resistant strains that appeared. It's because of the use of antibiotic in veterinary medicine. Of course, in Europe, the antibiotics are forbidden as growth factor, but they are still used in metaphylaxis or prophylaxis. The two famous examples for Salmonella multidrug-resistant strain are Salmonella typhimurium DT104, which is resistant to a lot of antibiotics. And this strain appeared in the 19th all over the world, except in Australia and New Zealand, because these two countries imposed a quarantine on imported animals. And more recently, another Salmonella multidrug-resistant appeared. It is Salmonella Kentucky. It comes from Egypt, where the antibiotics are massively used, and it settled recently in France. Besides the genetic acquisition of the gene, another thing is really important for microbial adaptation. It's bacterial stress and bacterial and stress response. In food processing environments, bacteria mainly live in a biofilm state on surfaces, and these biofilm protect the cells against heat treatment, dehydration, or disinfection procedure. Some cells can die, but some other that are protected can survive, and these cells that are submitted to sublethal treatments can respond to stress. They can produce more stress protein, they can modify their membrane fatty acid composition, or they can overproduce some exopolysaccharides, and then all of these stress response lead to an induced tolerance to subsequent stress, and then the bacteria can persist better in food processing environments. So we have here two examples of the pathogen persistence in food processing environment. The first one is about Salmonella, which was isolated in a processing environment of a peanut butter factory. And from this peanut butter, as this peanut butter was used as ingredient in numerous food, processed food in the country, there were a large-scale outbreak in the USA in 2009. So this outbreak was due to the persistence of Salmonella in the environment of the factory. Another bacteria that is really known for its persistence in the food processing environment is Listeria monocytogenes. One interesting example is this production plant of Turkey, and in this plant, from this plant, a sporadic case was appeared in 1988 in Oklahoma with the consumption of turkey sausage. And 12 years after, another outbreak appeared from the same production plants. This outbreak was much more large because it affected several American states, and during the investigation it was found that the strain was the same as the first one. So this strain of Listeria persists during 12 years in the same environment, despite the procedure of cleaning and disinfection. The second part is about change in microbial environments. So these changes generally lead to unusual transfer of contamination to food and to human beings. So there are some examples that I can take to illustrate these changes. The first one is about weather condition and climatic disorder. This weather condition can modify the survival, the growth, and the persistence of pathogen in environments. So, for example, it was shown that even seasonal increase in temperature can bring different risks. For example, it was shown that there is a 5 to 10% increase of salmonellosis per degree of ambient temperature, increase of ambient temperature. So it means that when the ambient temperature is higher, you have more chance to have salmonellosis. And moreover, in summer, the farm animals carry more pathogenic agent. Of course, when there are some extreme weather conditions, it will bring also new risk because of the water availability and water quality. During flooding, for example, the land and crops are submerged and the environment stays humid a long time, so the mold can develop and produce mycotoxin. Moreover, in this condition, in the wastewater treatment plants, the stormwater overflow are used to discharge the water in high quantity that comes to the plants. So this water that is discharged is not treated, so the safety of the water is not guaranteed. So sometimes you can have some selfish contamination with that. Regarding drought, it can also bring some changes in contamination of environment because it brings some migration of animals, some displacement of cultivation zone, and then all these displacements can bring some modification of ecosystem, and they can it can have some transfer of contamination from some areas that have high prevalence of pathogen to low prevalence area. So it can transfer contamination in low prevalence area. Another thing that is really important in these microbial environments is changes in agricultural practices. For example, the farm organization. At the end of the 20th century, there were a high increase of farm size, notably in USA, and notably in poultry sector. So, for example, in 2010, there were more than 2,000 salmonellosis due to shell eggs, and these shell eggs were produced in a huge farm that breeds more than 15 million of poultry. And these issues of contamination and spreading of contamination, because each time that one chicken is contaminated, the risk of spreading to all the farm is important, and so the consequences are high. And this type of problem, you can also have it in fruit and vegetable sector because the farms are also very big. Today, on the contrary, in Europe and specifically in France, we have more and more small organic and free-range farms. So these can also bring new risk because there were a study on more than 600 organic and non-organic pork farms, which demonstrated that there were a higher prevalence of Salmonella and Toxoplasma in organic farms. And moreover, Trichinella, which is a re-emergent parasite, has been detected in organic farm. So this change in size of the farm can bring new risk when it changes. Also, the slaughterhouse organization can have some impact because in the 20th century also the slaughterhouse size increased, and then the collection of living cattle from farm to farm can disseminate pathogen because the trucks that go from farm to farm can bring the pathogen to many farms. So it can be also an important way of disseminating pathogen. In agricultural practices, one thing also is important is the watering of crops. If the crops are irrigated with contaminated water, we can have some large outbreak. Two examples with raspberry, one with fresh raspberry which was produced in Guatemala, and they were contaminated by the parasite, the emergent parasite Cyclospora, and then several states of USA were contaminated, were affected by this outbreak, and these fresh raspberry were contaminated by the water. And also in Scandinavian country, it's quite the same story because it's frozen raspberry that were contaminated by the water containing norovirus. About the use of sewage sludge as fertilizer, it's important because in North America, it's allowed, they are allowed to use non-treated sewage sludge to fertilize the tomatoes, for example, the vegetables. And there were a large outbreaks because in the tomatoes, when the tomato plants and the tomato flower are contaminated by Salmonella, Salmonella can internalize into the fruit during its formation. So it's much more difficult to remove it after. It's not just a contamination in surface. Then about the mode of preservation, of food preservation. What is a practice that is evolving today is that the consumer demands much more fresh products that minimally treated, but with long shelf life. So it's possible to preserve non-sterile food products by contamination of hurdles such as mild thermal treatment associated with refrigeration and sometimes with packaging under vacuum or modified atmosphere. For this product, of course, you have to define a shelf life, a specific shelf life, to preserve the product. Of course, these products, these non-sterile products, are sensitive products to preservation failure. For example, a break in the cold chain. So this is not new, it's quite known. It's just enter in the mistake problems. But recently, we discovered that some sporulating bacteria can be psychotrophic. or sporulating bacteria are not killed by mild treatments, and then if they are psychotrophic, the refrigeration here is not enough to preserve the food. For example, some strain of Bacillus cereus are psychotrophic, and then they can grow in pasteurized vegetable purees that are preserved at low temperature. And another example is Clostridium botulinum type E, that is strictly anaerobic, able to grow and produce toxin at low temperature, and this bacteria can contaminate fish. So if the fish is preserved in vacuum and with refrigeration, the growth of this bacteria is not completely inhibited, because it can grow without oxygen and it can grow at low temperature. So Clostridium botulinum is an emergent risk in vacuum refrigerated fish products. Now about mode of consumption. We know that nowadays consumption of exotic food products expand in Europe, notably some raw food. The fish is a good example. The fish is generally cooked, but more and more we consume the fish raw in the well-known sushi and sashimi, of course, but also with ceviche from Peru or marinated herring and anchovy. So in raw fish, there is two main microbiological risks. The first one is a parasite, which is called anisakis. This anisakis is the final host of anisakis is marine mammal. So from the marine mammal, the larvae are disseminated in water, and from the water, the fish are eating the larvae, and when the fish dies, then the larvae goes from the abdominal cavity to the flesh of the fish, and then the consumer eats it and can have a gastroenteritis, gastroallergic symptoms, or skin allergy. So this anisakis is found all over the world in sea and ocean, but the risk with farm fish is near zero, because there is no mammals around the farm fish. So as the final host is not present, the risk is near zero. But for wild fish, the preventive mean, except cooking, of course, because cooking is killing anisakis, but except cooking, we can have other means for avoiding this intoxication. We can have early evisceration, because it will prevent the parasite to go to the flesh of the fish, and we also have a visual inspection, because we can see the larvae if it's not too small. But it's not completely efficient, these two preventive means. The only one that is really efficient is the fish freezing, and this is mandatory to sell raw fish product in Europe. You have to freeze the product at -20 at least during 4 days. But freezing is not efficient against bacteria. So the second main risk in raw fish is Vibrio parahemolyticus, which is a bacteria that contaminates fish and sea environment, and it cannot be destroyed by freezing. So this a highly virulent serotype of Vibrio parahemolyticus, the O3K6, was the origin was in the Southeast Asia, and then it goes to USA, and then to Chile, and then to Spain. All that because it is transported by commercial boats. These commercial boats use ballast tank, and the water is transferred from one estuary to another. So now this bacteria is present in many estuaries in all the world. This bacteria is allophilic, so it's perfectly adapted to the sea environment. It can survive a long time at very low temperature, and it's often associated with a wide variety of seafoods. It, of course, can be destroyed by cooking because it's not spoilating, but not by freezing. So if we want to eat some raw fish, the only thing we can do is to have a short shelf life in raw fish product and to store the product below 5 degrees. Because this bacteria is really virulent, notably against immunocompromised patients, because it causes sepsis. So it's really virulent. And we can see here the estimated percentage of change in the incidence of infection. And we can see that here, the incidence of Vibrio infection in USA increase much more than the other foodborne pathogen. You see that Salmonella is decreasing, but Vibrio is increasing, and it's mainly due to consumption of raw fish. The last example I will take to illustrate the change in microbial environment is about trade and exchange. The dissemination of pathogens all over the world is increasing through the globalization of exchange. Exchange of people, exchange of goods, exchange of animal and vegetable raw materials, and the exchange is done between exporting and importing countries that not necessarily have agricultural and hygiene practices, the same agricultural and hygiene practices. So if the practices are different, the monitoring of pathogens are different, and then it's we can bring some pathogen from a country to another. Moreover, the regulation for the use of antibiotics are also not the same, and the efficiency of monitoring and pathogen detection can also be defective in some producing countries. So this brings the risk to disseminate some pathogen in all over the world. So formerly, the outbreaks were localized in the area of food production, but now there is a large increase of falsely sporadic case, because the cases have the same origin, but they are dispersed all over the world. So some example of worldwide outbreaks and dissemination of pathogen. The first one is about exotic fruit commercialization. The consumer wants exotic fruit all the year. So there are massive importation of fruit from producing country. And for example, in 1999, there were an outbreak that was that affected 13 American states, and the serotyping of the salmonella, the involved salmonella, showed that all the outbreaks in all the American states were of the same origin, and the origin was imported mangoes from Brazil. The problem was that these mangoes were dipped into water to avoid the fruit of the fly of the fruit, And unfortunately, this water was contaminated by salmonella, and salmonella internalized into the mangrove and then caused this outbreak. The same thing was done with the parasite, the intoxication with raspberry in Guatemala with the parasite Cyclospora. It spread in several American states because this raspberry was commercialized all over these American states. Besides exotic fruits, cattle transportation can also be a worldwide dissemination pathogen. The examples I gave you of Salmonella typhimurium DT104 that appeared all over the world nearly at the same time because the cattle was transported all over the world. And the only country that were not affected by these bacteria were Australia and New Zealand because of the quarantine. And of course, also the serotype, the virulent serotype of Vibrio paramolyticus, which was disseminated in all history or a lot of history in the world by the commercial boats. So these examples show that as soon as a pathogen appears somewhere in the world, it can be disseminated all over the world by the transportation, the commercial transportations. So the last, the third and the last categories of factor that bring some new risk is changes in human susceptibility. We know that susceptible people are infants, pregnant women, the elderly, and immunocompromised people or patients with chronic disease. The three last increase. We know that these three increase because of the progress of the medicine. For example, we can see here the life expectancy increase between 1951 and 2013. And a woman, for example, gained 16 years in this period. So we see that there are more and more old people. And old people have less efficient immune system, and also some other defense system that can be altered. For example, the acidity of stomach. So these old people are more susceptible to foodborne disease, just like immunocompromised people and patients with chronic disease. So we have seen in the last year emergence of pathogens that are particularly virulent against these susceptible people. It's the case for Listeria monocytogenes. It's the case with Vibrio paramolyticus or vulnificus that can the lethality against susceptible person is really high, and also Chronobacter sakazaki that cause sepsis in infants. So we can see here in this table that the old people over 65 years have 7.5 are 7.5 more susceptible than a control adult. And you can also see in the same table that a person having pulmonary cancer or bladder cancer or have been transplanted have a high susceptibility, which can reach more than 2,000 times compared to the control adults. So we can see that these people are much more susceptible to Listeria monocytogenes. And moreover, it has been shown in a study that the elderly are even susceptible, more susceptible to classical foodborne pathogen like Salmonella, for example. They showed that the fatality rate of salmonellosis for elderly living in a retirement home is about 7%, and we can compare this rate to the rate in the whole population that is really lower because it's about 0.5%. So in conclusion, I would say that microbiological hazards are evolving. So when a new infection disease appears, we have to find quick solution to detect, to identify, to characterize the new microorganism and or its new mode of transmission. And this is important to be able to propose quickly some relevant control measure. So for the characterization of an emerging hazard, it's necessary to look at several points of view. The first one is a clinical point of view in order to treat the sick person. So what is the disease? What is the severity? Is the bacteria or the microorganism resistant to antibiotic? Is the treatment available? The other point of view is an analytical point of view, because we have to have a quick detection of the bacteria and to propose quickly control measure. So the question is there a detection method in food or in blood for patient? Are these methods exist? And what are the characteristics of the bacteria, the growth characteristic and the resistant characteristic? The last one is the epidemiologic point of view, because it's important for evaluation of the impact on the whole population. What is the infection occurrence? What is the reservoir and the mode of transmission to human? What is the infectious dose? And what is the incubation period? So now I think that for to better manage food safety, we have to find a multidisciplinary approach to be able to increase the efficiency of surveillance and to better understand the mechanism of emergence. The first thing is that we can use technology for real-time monitoring. There were demonstrated in other sectors than food safety, but they can be applied in food safety, notably to monitor the exact temperature of the product. Also, the genomic technique can help, because they can give information of for identification of pathogen, or they can increase the speed of detection. Moreover, the predictive modeling can be also interesting for understanding microbiological growth, epidemiology, dynamic of infection disease, and also for attributing the source. What are the main food products that are contaminated but by these emergent hazards? So the source attribution is really important. And then at the end, of course, the risk assessment. But anyway, the consumers has to be aware that zero risk cannot be achieved. So improving the food safety means reducing the risk, not to zero, but reducing the risk and increasing the reactivity in case of epidemic. So this is the reference for my presentation, and I thank you for your attention. So I wait for your question.
Thank you very much, Florence, for this really nice overview. I'm sorry that I realized that my presentation was not shown, but I already wrote in the chat box that everybody can download from the handouts, so you can see it later. About this presentation, are there any questions now? So you please, if you want to ask a question, please raise your hand or type something in the question box. So there is a question from Ainoa. I think the hand was down, but Ainoa, do you have a question? Then please raise your hand again. No? Maybe I can start with the short questions. Maybe you have an answer. I don't know if you have data, but a topic in the food industry is the cleaning and disinfection, and there's also the problem of the resistance. So the bacteria, microorganisms adapt to these disinfectants. Yes. Do you have data if this is increasing or the resistance?
We know that bacteria can adapt, and we have data showing that the bacteria growing into biofilm are much more resistant than planktonic bacteria that are used to test disinfectants. So we know that these resistance can be over 1,000 times compared to planktonic resistance. So yes, the persistence of the bacteria in the food processing environment is mainly due to these bacteria living in biofilm, and this biofilm protecting them.
Mm-hmm. Maybe in the meantime there is another question. Or maybe it can also a contribution if you have some data or research you want to show. It's also possible that you show some slides or anything else, or a website which is interesting maybe for the other participants. So there is only one comment. I don't know if you see the comment, Florence, from Teresa Gonsalves. She is thanking for your clear presentation.
No, I cannot see the comment.
Maybe I have to forward this to you. Okay, then you should see. So there is this was the question I already forwarded. Mhm.
Vielleicht liest du die Frage vor, weil die anderen sehen sie nicht.
Yeah. Yeah, it was not a question, it was just a comment that she was thanking for the well-explained webinar, and it was interesting to hear the risk that globalization can bring to food safety.
Mm-hmm. Okay.
So there is another question from Linda. Oh, it's also just a congratulations to you. Okay. So you don't need to be shy, because it's really a good experience or a good chance to talk to an expert in this field. So if you have any questions, just ask. You can type, or you can also raise your hand and I open your microphone so you can speak. There is a question from Arthur Roig. Maybe I open your microphone and then you can. Yeah. So this is not. Why can I not unmute? So Arthur,
hello.
Yeah, hello.
Can you hear me?
Yes, very good.
Okay. First of all, congratulations for the presentation. It was extremely interesting for me. And I have one question that I write it in the chat, and it's just that if I wonder if the mechanism that induces the resistance to antibiotics, if these mechanisms are similar to that induces the resistance to disinfectants. In other words, are the bacteria that are more resistant to antibiotics at the same time more resistant to disinfectants or other adverse factors such as physical factors, etc.?
Yes.
Is there any information available about that?
Yes, it's because both. Because some mechanisms are the same. Because for example, if the membrane became, because the bacteria is stressed by the antibiotic or the disinfectant, and then the membrane goes to be more rigid, and this can induce cross resistance between antibiotic and disinfectant because it's non-specific mechanism of resistance. So that can exist, and so it is described in the literature. But some other mechanism of resistance to antibiotic are much more specific. For example, when some bacteria acquire some gene coding for efflux pump to one specific antibiotic, then it's not a cross resistance with disinfectant, because disinfectant will not have the same mode of action. So it depends. I said sometimes there are cross resistance, sometimes not.
Okay, thank you very much.
And there are. It's a really good question, because we are going to work in my laboratory. We are going to work on that topic, because it's a really good question to know what are the interaction between the antibiotic treatment and the disinfectant procedure. Because on the contrary of what I present, because I present the antibiotic resistance, and does it bring disinfectant cross resistance? But the contrary can be also interesting. If we use disinfectant and if this bacteria goes to adapt very much to the disinfectant, then when the bacteria goes to human being, are they resistant to antibiotic? And that is a point that we are going to study. So it's some evidence starts to be demonstrated, but it's not completely well known. So there are some studies that will be focused on that in the future.
Okay, thank you very much.
Thank you also. We have 3 more questions now. Very good. So first, maybe there is just an oral question, and then we have 2 written questions. So first, I would like to ask Christos Apostolopoulos from Greece. I will open the microphone.
Hello. Can you hear me? Thanks. Congratulations for the presentation. Thanks very much for this. My question lies on the existence of quite a lot of natural antimicrobial substances that occur in various foods. Can you see any use of that, and what degree of protection we can achieve by combining those together?
Yes, that's a topic also we are working on in my laboratory on natural antimicrobial, notably the one that are extracted from aromatic plants. And we have shown that some of these natural antimicrobial are efficient against bacteria, to destroy bacteria, but also to destroy biofilm. And some can be really interesting. And we had we studied some hydrosol of essential oil together with a Greek university, and we have demonstrated that this hydrosol of essential oil was really active, really efficient against biofilm because it penetrates really efficiently into the structure of the biofilm. So yes, there can be some application of these natural compounds, but unfortunately, we have some problem also of in food. We have some problem of regulation because these compounds are not allowed in the European regulation. We cannot use this natural antimicrobial as antimicrobial in food products. But in food processing environment, it would be much more easy to use.
Okay, thank you.
Thank you very much. There is also a contribution from Teresa Gonsalves. Maybe I open also her microphone that she can talk to us, if possible. Teresa?
Hello.
Hello.
Hello. Thank you very much for your webinar. And it was very interesting to hear the new challenge to food safety. And well, my question is not only about this issue, but what concerns us. There are a lot of around the world, and for me, it is important to.
I'm sorry, I can't hear you. I can't hear you. I didn't hear the question.
Well, there are a lot of legislation on food safety around the world. Can you hear now? No.
I hear you, but it's not clear. So I just try to guess what you say, but can you write it down?
Yes, I write it.
So I will forward the question to you, or maybe I can. You can hear me, Florence?
Yes, I can hear you. Yeah.
I forward these questions to you, then you can read, but I can also read for you.
Yes.
So there are two contributions. The first one, is it possible in the next future to consolidate all legislation on food safety and spread it as a rule for all?
All over the world, you mean?
Yes.
Because the food regulation is yes, for European country we have a regulation for European country, but that's true that for antibiotic, for example, the question is really good because in Europe we are not allowed to use antibiotic as growth factor, but in some countries in Middle East countries or in America, for example, they can use these antibiotic with the different regulation than we have in Europe. So that's a problem because pathogen will not have no frontiers, so they can go and disseminate all over the world. So you are true, the legislation has to be the same everywhere to avoid this increase of antibiotic resistance.
Yes, that's my question. Thank you very much.
You're welcome.
There was also the second part of your question. She was writing that of course, it's difficult to bring all partners on a table and to agree what is the best, but as food safety is a concern for all in the world, and also the microorganisms can travel, also good practices can be spread. Yeah.
Yes.
Okay, then we have another question by Amin Musabi Kaniga. Maybe I open his microphone. I have to find in the list. Is Amin? Okay. Amin, are you here? Amin, do you want to speak? Your question was also about regulations. If there is any movement regarding to update the regulation against newly formed problems. Do you have any idea?
Against, what did you say? Against? Can you repeat the question?
Yeah. Is there, what about regulations? Is there any movement regarding to update the regulation against newly formed problems? Do you have any idea?
Of course, the regulation has to take into account the new pathogen. For example, E. coli, enterohemorrhagic E. coli was not such. It was not mandatory to analyze to search for enterohemorrhagic E. coli in seeds, for example, before the 2011 outbreaks, and then now it's mandatory to search for this pathogen, because we know that it appeared in this type of product. It's the same for Conobacter sakazaki. When it appeared, the regulation changes, and then now in milk powders we have to look for a Conobacter sakazaki because of the infection that have been that have emerged with this bacteria. So the regulation has to change when the new microorganism appear, but it can take time.
Does this answer your question? He apologized, he has no microphone, but maybe you can type if this is what you expected as answer. Amin? Okay, thanks. Is there any further question? So it doesn't seem there's no further question at the moment. So with this, I would like to thank all the participants and also especially Florence for the presentation and the participants for listening and attendance and also for their contributions. And I will close this webinar, and hopefully we will see on another webinar. You can see the list of future webinars on the FooD-STA website, and there will be in the future there will be also different moderators. The next moderator is sitting beside me, Christine. Maybe I can introduce her from LVA. She will moderate the next webinars. Not the next one. The next one is moderated by somebody else, but the future ones. So thank you very much, and you will receive also an email afterwards, also with a link to the recording. So thank you very much. Bye-bye.